Hello, my name is Dr. Sue Whittle and I'm a senior lecturer in biochemistry at the University of Leeds. In this podcast, we're going to look at the way in which living things metabolize glucose to provide energy. There are basically 4 sections here. In the first, I will look at some useful background information, such as oxidation and reduction reactions, and the form of energy that living things use. In the next 3 sections, we'll consider how glucose is metabolized to produce energy in the form of ATP through glycolysis, the Krebs cycle, and oxidative phosphorylation. Every living organism needs energy. It uses this energy for a wide range of different purposes, such as movement, growth, and replication. Looking at this from a molecular perspective, energy is required for synthesis of new molecules, such as DNA and proteins, to drive some enzyme-catalyzed reactions and active transport, and a whole host of other purposes. All organisms can obtain energy by metabolizing glucose. This process is known as cellular respiration. We have all seen the equation for obtaining energy from glucose in the presence of oxygen, producing carbon dioxide, water, and energy, as is shown here. This is an example of an oxidation reaction. And before we go any further, we need to talk a bit about oxidation and reduction reactions, as this concept comes up regularly during the rest of this talk. There are several ways to define oxidation and reduction. At this level, we think about this process in terms of the transfer of electrons. In an oxidation reaction, electrons are lost, while in a reduction reaction, electrons are gained. You'll remember this as oil rig from your GCSE science. Oxidation is loss, reduction is gain. We refer to these reactions as redox reactions because they come in pairs. If one molecule is oxidized, then another is reduced. In this talk, we'll come across a series of very important redox reactions involving a coenzyme or cofactor called NAD. A coenzyme is a molecule which helps an enzyme to carry out a reaction, usually by donating something the reaction needs or by removing something that needs to be got rid of. These are often derived from vitamins in our diet. The role of NAD is to transfer hydrogen and electrons from one molecule to another. The NAD becomes reduced and the other molecule in the reaction becomes oxidized. On this slide, you can see an example. In this diagram, you can see a reaction which is very important in cells which are starved of oxygen. We'll talk about it again later. In this reaction, pyruvate is converted to lactate or lactic acid by the enzyme lactate dehydrogenase. Just to avoid confusion, it might be worth saying that in biochemistry we tend to use shortened forms for the names of common chemicals involved in metabolism. So we often use lactate for lactic acid, citrate for citric acid, pyruvate for pyruvic acid, and so on. Looking at this slide, you will see that this reaction is reversible, like many biological reactions. If we look at the reaction going from left to right, i.e. the synthesis of lactate, we can see that the enzyme uses reduced NAD or NADH to reduce the pyruvate. The NAD is then oxidized. When lactate is converted to pyruvate, the opposite happens. The NAD is reduced to NADH and the lactate is oxidized. If we break this reaction down a bit further, we can see the electrons being transferred from one molecule to another. The NADH loses hydrogen atoms. You can think of these as splitting into hydrogen ions and electrons. So when pyruvate is reduced, it gains 2 hydrogen ions and electrons which came from the NADH. Now, back to glucose. In living organisms, the glucose is broken down slowly in a series of small steps, and the energy created is in the form of adenosine triphosphate, or ATP. Before we look in detail at ATP synthesis, it might be useful to think about why ATP is a good form of energy to produce and how living things use it. Remember, many reactions don't happen spontaneously. They need energy to get them started. You saw this in the video. Glucose won't react with oxygen if we just put it on the lab bench in the air. Energy from the heat of the Bunsen burner was needed to get it to react. The same is true in a cell, but of course in a living organism you can't heat up the components to get them to react, because heat would destroy the organism. Living things use enzymes to decrease the amount of energy needed to get the reaction going. This is called the activation energy. But sometimes this on its own isn't enough, and a safe source of additional energy is needed, and this is where ATP is used. This slide shows the structure of ATP. It consists of a molecule of adenosine coupled to 3 phosphate groups, hence the name, adenosine triphosphate. The most important part of this molecule is the bond which holds the final phosphate on to the rest of the molecule. For the chemists amongst you, this is known as a phosphoanhydride bond. This bond can be broken by a reaction called hydrolysis, producing adenosine diphosphate and a free phosphate ion, often written as Pi. Sometimes the phosphate is released, sometimes it's added to another molecule. We will see this happen when we look at glycolysis. As you probably know, when chemical reactions occur, energy can be released. And when ATP is converted into ADP, a considerable amount of energy is released. This energy can then be used to make reactions happen that wouldn't otherwise. For those of you who are also doing chemistry at A-level, you might want to think about why the conversion of ATP to ADP produces so much energy. ADP is a much more stable molecule than ATP. Can you see why? Look at all those charged oxygen atoms held so close together. Imagine that a cell wants to convert molecule A into molecule B using an enzyme. This reaction requires energy to make it happen, so on its own, no reaction will take place. However, if a molecule of ATP is hydrolyzed at the same time, the enzyme can use the energy released from the ATP conversion to drive the conversion of A into B. This is called a coupled reaction. To maintain the ATP level in a cell, the ADP formed when ATP is hydrolyzed then needs to be converted back into ATP. So, how do living things make ATP from ADP using the energy from glucose? This happens in a number of steps, which we can break down into 3 linked processes: glycolysis, the Krebs or citric acid cycle, and oxidative phosphorylation. If you come to university to study a biological sciences subject, you'll have the opportunity to learn about every reaction in this vital process, and perhaps even more importantly, how those processes are controlled. But for now, the most important thing is to get an overall sense of what's going on and where in the cell each set of steps happens.